Thanks, Patty, and thanks everyone for your interest in this topic. It's my distinct pleasure to really introduce Michelle Mielke, who is a professor in the Department of Health Sciences, Research Division of Epidemiology, and a professor in the Department of Neurology at the Mayo Clinic in Rochester. She and Ron Peterson and Dave Notman and I have done many different kinds of projects together, so it's really a delight to host her here in our home. Michelle has received her bachelor's degree of science in neuroscience at the University of Pittsburgh and a doctorate in neuroepidemiology at Johns Hopkins. And she works predominantly in what I think is a burgeoning field of translational epidemiology. And it's to further understanding of the etiology and epidemiology of neurodegenerative diseases. But another big focus, and why she's here today, is her research on understanding the sex and gender differences in the development and progression of neurodegenerative diseases and other aging-related conditions. So it's my distinct pleasure, and I hope you all give her a very warm welcome, to introduce to you Professor Michelle Mielke.
All right. Well, thank you so much to Doreen, and it's a pleasure for me to be here today, and I've had a lot of great conversations. Let me see if I can figure this out now. So I know we have quite a few people, but if you have a burgeoning question or you want to refute what I'm saying and completely disagree with it, go ahead and jump up and interrupt me. So today I'm going to talk about sex and gender differences in Alzheimer's disease, whether they exist and if they matter. These are my disclosures. So I'm going to primarily give a broad overview of the field, including the epidemiology of sex differences, incidence, prevalence, how some of these sex differences might come around in terms of differences in brain structure, and then some possible differences in terms of risk factors, both sex differences as well as sex-specific. And with a lot of this, I hope there's a lot of questions that we don't have answered yet. And so my goal today is just to try and get you to think about this a little bit differently and to understand why we need to look at sex differences in all of our neurodegenerative diseases. So before I start out, I just want to make sure that we're all on the same page with basic definitions. So sex and gender is often used interchangeably, but really they're two separate things. So sex is when you talk about the biological constructs, so the XY chromosome related to gonads and hormones, estrogen, testosterone. In contrast, gender is the societal and legal aspects. Now, you can't completely separate gender from sex. Gender is often rooted in sex, but it has different connotations in terms of society and legal. So historically, women have had less access to education, less access to jobs, less physically active, at least in 50s and earlier. There may be differences for access to medical care between men and women. And even if you think about in Saudi Arabia, I think this past year was the first time that women have been able to drive cars. So that gives you a little bit more of a sense of what I'm talking about with gender. I also want to be really pedantic in terms of epidemiological terms too, because how many here have heard that women are at greater risk of Alzheimer's disease? Probably the vast majority, and it's in a lot of headlines, New York Times and whatnot. But it really depends on how you define that. And so I want to point out that nuance today. So when we talk about frequency, we're talking about the number or count. So if we're talking about the frequency of Alzheimer's disease among everybody that's diagnosed with Alzheimer's, we want to know the frequency of women, how many women of all are Alzheimer's patients. When we talk about the prevalence, we're actually comparing, say, women with Alzheimer's disease compared to all women, that proportion to men with Alzheimer's disease compared to all men. And the reason that's important is because, of course, women live longer than men, and so there's more women at older ages, and age is the biggest risk factor for Alzheimer's disease. In contrast with incidence, again, I realize this is very basic, but really we're focused here on if we're comparing a man and women in terms of sex differences at the age of 80 and we're following them up for 5 years. So is a man or woman more likely to develop Alzheimer's disease during that time frame? So question first, are more women affected by AD and are women at greater risk? So when we look at the frequency, I think it's well known that the burden of Alzheimer's disease is more on women compared to men. In terms of about roughly 2/3 of those with a clinical diagnosis of Alzheimer's disease are women. So the frequency is certainly higher for women than men. But when we start to look at the prevalence and specifically the incidence, it's not so clear. So this is a study, a review, let me see. Okay. That was done in 2002 and highlighting some of the differences in terms of incidence across a variety of countries. And what's really striking here is that you can see that in European countries, so Sweden, UK, and France, as well as the Europe pool data, and this was the Eurodem data, at older ages, women were at greater risk or greater incidence of Alzheimer's disease compared to men. However, when you look at the Framingham study and studies that we've done in Rochester, Minnesota, again, this was close to 20 years ago, we didn't see a sex difference at those upper age ranges. So since that time, of course, there have been a lot of additional incidence studies throughout the world. And there are really very few studies that are showing that women are at greater risk, including at older ages. The only study in the US that has found this was the Cache County study from Cache County, Utah, with an increased risk after 85, and the Rotterdam study showing an increased risk after the age of 90. But there's a whole host of other studies that are not finding any differences at all. Interestingly, a couple years ago, the Canadian government had requested reviews from a variety of neurodegenerative disorders. And so they did look at sex differences in these, and the systematic review that came out of 22 studies, neither the prevalence or the incidence significantly differed by sex when they combined all the data. There was a trend, but it wasn't statistically significant. I think the p-value is like 0.4 or 0.5, so it's not that it was close either. And then after that meta-analysis, there was another meta-analysis from Canada with studies around the world. There was another meta-analysis that came out looking at specific European studies. And that study, again, found that women were at greater risk of Alzheimer's disease compared to men, just looking at European studies. So what about changes in trends over time? Everybody's probably heard of studies that have suggested that the risk of Alzheimer's disease or risk of dementia is declining over time. How does this compare for men and women? Well, one of the first studies that looked at this was the Cognitive Function and Aging Study 1 and 2. And this was a study in the UK. They did their first incidence wave between 1989 and I believe it was 2002, and then the second incident wave 2007 to 2011. And there's a couple things that's specifically striking about this study. So one, in the first incidence wave, as we're looking at men here, and the blue is the first wave, the purple is the second wave, you can see that actually in this study in the UK, men had a higher risk of developing dementia than women did, which is kind of odd. But then over time, interestingly enough, the incidence decreased in men, whereas the incidence remained about the same or actually increased in women during this time period. In contrast, when we look at the Framingham Heart Study, we actually see more of a decreased risk for women as compared to men over the various epochs. So I'm throwing a lot of data out at you, and it's not agreeing all that well. So there's a lot of disparities in this, and what are some of the potential explanations for some of these disparities? So one is that there are potentially smaller sample sizes at upper age range. So there are a few individuals, men or women, that are enrolled that are 85, 90, 95 years old, and that's where we're seeing the major changes, at least in the European populations. So possibly unstable estimates. But it's also interesting to think about at these upper ages, when you talk with Claudia Kawas or Maria Carrata from the 90-plus study They tell me that really there are no sex differences in pathology at the upper age ranges. And one of the main reasons for this is that at that age range, you're not going to have anybody with pure pathology. Everybody has mixed pathology. And so it's really hard to discern what some of those sex differences might be. Another possible reason for disparities, particularly between Northern Europe and the US, is the use of different diagnostic criteria. And it's not that it's completely different, but there may be subtleties. So I did some of my thesis work with Igmar Skoog in Gothenburg, Sweden. And when we were doing the work at the time, I was interested in vascular risk factors for Alzheimer's disease. And the way they were diagnosing Alzheimer's disease is if you had a previous stroke, regardless of whether it seemed to affect cognition, you couldn't just be an Alzheimer's case. It would have to be mixed dementia. And I know, others, if that would be put into that would be considered Alzheimer's disease. So there may be some of these subtle differences that could be causing the differences. Third is the effect of differential mortality. And in fact, the Framingham study found that the lifetime risk was higher in women only after correcting for competing risks of death. And this is something in epidemiology that we haven't fully done a good job of exploring yet. Women live longer than men again, so women have a higher burden of all age-related diseases. But we do need to consider differential mortality in considering risk factors and risk of these diseases. And then lastly, another potential explanation for disparities, particularly between Europe and us, are social, cultural, and historical events. So I worked very closely with Walter Roca, who was involved in some of the initial Eurodem studies. And it's interesting to listen to him talk about World War II, the effects after World War II. And his hypothesis is that, in terms of both women and men, there were differences experienced whether you were in Europe or whether you were in the US. Now, a lot of men were fighting over in Europe, and of course they have a different experience, but women in the US had a completely different experience than women in Europe did over World War II. And even afterwards, with the changing of governments, the establishment of governments, a lot of different changes, there were different stresses and whatnot. And so that could potentially be another reason why women are at higher risk at some of the older ages in European countries compared to here. So then there's also differences in terms of education. So during World War II, women were less likely to get education, had less than men. And so, one of the questions coming up, and just in general, is that with the increase in education, particularly for women, will that affect their risk of dementia and possibly sex differences down the road? So I think, when we look at the incidence, yes, the burden is greater for Alzheimer's disease, but I'm very leery about coming out and saying that the actual risk is greater for women. And I'll touch on this a little bit more. I think it causes excess worry at some points. The problem is when you're talking about sex differences, the main reason to look for sex differences most of the time is that an incidence or prevalence is higher in men versus women. And so as I've given talks on this and come out and say, well, look, in the US, we're not seeing a higher incidence of Alzheimer's disease in women, then I will get responses of, well, then why are you looking at sex differences? There's no reason to look at sex differences. But honestly, there's absolutely reasons to look at sex differences. And so what I'm trying to get people to understand in the talks is that, when we look at cardiovascular disease, we know that there are different risk factors for men and women. We know that there are different ages of onset. We know that there are different treatments, different preventive measures. Yet cardiovascular disease is the #1 killer for both men and women. And so I think we need to move beyond this idea of incidence and that there might not be a difference, and really just focus on sex differences, because I do think that there are sex differences in terms of the pathologies, the clinical presentations of Alzheimer's disease, and possibly down the road treatments. So for some examples, we already know that there are clinical differences. So one is that the risk of MCI is generally higher in men. We found this in the Mayo Clinic study of aging, and it's been found in other studies as well. But the transition from MCI to dementia tends to be faster in women. One of the possibilities for this is that the primary measure that's typically used for memory measures to test for whether somebody's developing dementia or MCI is verbal learning memory measure, and women do better on verbal learning than men. And so Pauline Mackey and Aaron Sunderman and others are looking at the sex-specific cut points, and that may be why women are less likely to have MCI and to progress faster than men. But there's a lot of work that still needs to be done there. Secondly, there are more and more studies suggesting that the behavioral symptoms that are common and typical of Alzheimer's patients are different in men and women as well. So anxiety and delusions in a couple studies now have been found to be more common in women. In contrast, apathy and agitation has been found to be more common in men. And there's a nice study that came out earlier this year that replicated these differences, but also suggested that women in general display a broader range of symptoms and have more severe symptoms on average. Lastly, there are some indications that there may be sex differences in response to treatment. Now, again, these aren't disease-modifying treatment, but anticholinergics may have symptomatic benefits. It's well known that sex hormones exert tropic effects on the cholinergic system, and we have previously shown that there were some sex differences in terms of cholinesterase use and cognitive decline among Alzheimer's patients. So again, if there isn't an incidence or a difference in incidence, what are some sex differences in brain structure or brain function that could be contributing to some of the differences we see or to some of the different pathological the pathways? Well, it's well known that men have larger head sizes than women do, and they also have larger cerebral volumes. And so this has brought up the question of whether men can withstand more pathology versus women. And the Ross MAP study, Lisa Barnes, had shown that women have a higher odds of clinical diagnosis of Alzheimer's disease, even with the same pathology as men. So is it because of their smaller head size? They have less resilience? That's still unclear. But interestingly, among cognitively normal individuals, we have found that men have faster age-related brain volume decline compared to women. So this could be a sex-specific change that is very disease-specific. Also, women have a higher percentage of gray matter, but men have a higher percentage of white matter. So how might this actually affect various processes and risk of developing Alzheimer's disease or other neurodegenerative conditions? And I think this paper that came out by Katani is a good example of things that we need to consider. So they had looked at language lateralization and sex differences. And when they started out, when they combined all the study, they found that about 62% of individuals, so the vast majority, had a strong left lateralization. There was a group 2 of about 20% that had bilateral but left predominant. And then there was a slightly smaller group that was bilateral but symmetrical in terms of both left and right dominance. Next, they looked at sex. And what you can see is that for men, the vast majority were in group 1, strong left lateralization. Much fewer down, and especially in terms of being symmetrical, much fewer men. But the women were very similar across all these groups. And so there's a lot more variability in terms of the structure for women and white matter pathways. And it's interesting, although this is language, it is interesting to think about how these other pathways might predispose a man or woman to Alzheimer's disease down the road. And I think this is something that hasn't been looked at enough in the field to date, and something that we need to certainly look at more. Another thing to think of in terms of brain structure and function is chromosomal activation. So a lot of times you think of XY chromosomes or people that I talk to, and it's strictly more hormonal basis. But there are really a lot of genes on the X chromosome in addition to being important for reproductive factors and hormones. So specifically, if you look at the brain, there are genes on here that are associated with mental retardation, social cognitive function, cerebellar ataxia, and even color blindness. What's interesting is that beyond just all the genes that are affected here, but women's are mosaics of X. And so there's within each cell, there will be one X will be inactivated. And so within a specific tissue, you could have cells, one with a paternal X and one with a maternal X. And it's not clear how any of this might relate to brain diseases. There was a study a few years ago, and I haven't found, if anybody knows of another study, please let me know. I haven't found of another good study that has looked at X inactivation in the brain. But this is a nice example where the green, I think the green is paternal and the red is maternal or vice versa. But they labeled the various X's, and you could see the distribution throughout the brain. Now, in this particular case, it's very lateral. I don't know if it's going to be like that for most tissues, or if this is typical in the human brain. But it starts to make you wonder in terms of paternal and maternal inheritance, what that effect could be in terms of risk of neurodegenerative diseases. So how about differences in AD or cerebrovascular pathology? So when I talked about the incidence earlier, that was based on general clinical diagnosis. And of course, we know that some individuals diagnosed with Alzheimer's disease don't have amyloid in their brains. They have other contributions and vice versa. So we and others, I think a big topic in the field right now is looking at sex differences in terms of pathology. And this is a study worked on in collaboration with Cliff Jack, looking at both amyloid, tau, and neurodegenerative abnormalities within our population in Olmsted County, Minnesota, so Rochester, Minnesota. And the benefit of this is that with using the Rochester Epidemiology Project, we're able to follow people between different hospitals and different medical facilities. And we randomly sample individuals into our Mayo Clinic study of aging. And so we know the diseases of those individuals that don't agree. We know the diseases of those that agree. So we can upregulate and actually have a population-based estimate of how many people have amyloid, how many people have neurodegeneration, and how many people have tau. And in general, what we found was that the associations between each of these biomarker positivity groups were very similar between men and women. There's very little differences. There are a couple differences. So this purple is the amyloid positive, tau negative, or amyloid positive, tau positive. So amyloid positivity is a little bit higher in women compared to men, wasn't statistically significant. Neurodegeneration positivity was a little bit higher in men compared to women. But overall, we didn't see an exceptional difference. We've also looked in the CSF, CSF amyloid, CSF t-tau, and p-tau, and we are also not seeing a difference in terms of markers there. I think there have been other studies out recently that are also not finding a difference, or if they find it, it's very subtle and not statistically significant. There have been a couple studies out recently that suggest women who are amyloid positive are more likely to go on to progress and become tau positive compared to men. But I think there's a lot more work, again, that's subtle, and there's a lot more work that needs to be done. So it's still possible something there, but there's not a glaring sex difference in terms of the pathology. I also wanted to point out there, Melissa Murray down in Mayo Jacksonville did a study in terms of looking at overall brain pathology. And they were interested in looking at the clinical diagnosis and comparing it to the pathological diagnosis. So they had close to 1,600 autopsies with confirmed AD from the Florida Brain Bank, and looked at amyloid pathology, neurofibrillary tangles, also looked at cerebrovascular disease. And in general, what they found was that there was overall similar frequency in tangle burden as well as amyloid pathology for men and women. But when you start to look at it by age, there were some subtle differences in terms of diagnosis. So they found that prior to the age of 70, men were less likely to be diagnosed with Alzheimer's disease, even though they had Alzheimer's pathology, whereas women at older ages were slightly more likely to be diagnosed with Alzheimer's disease, even though they typically had mixed pathology. And one of the reasons they think this is that there was a higher percentage of hippocampal sparing AD among men. So they were more likely to have potential more atypical features, maybe first start out with executive dysfunction as opposed to memory at some of these younger ages. And so as a result, they're not showing the memory problems, and so they're not being diagnosed with Alzheimer's disease. In contrast, women had a higher percentage of limbic predominance, which of course is going to be more associated with some of the memory phenotypes. And so they're also more likely to have a typical AD-like clinical presentation. So a couple things from this study. One, suggesting again that the pathology doesn't differ by sex. Two, that there can be different clinical presentations for men and women. And three, I think, in terms of diagnosis and this idea of, with all the headlines of women are at greater risk, it may be detrimental to women down the road, well, both women and men, in terms of, especially if you're this is a fantastic institution in terms of diagnosing specific types of dementia, but the vast majority of dementias are going to be diagnosed in primary care physician offices. So if you see all the headlines of women are at greater risk and they come in and say, I have a memory problem, well, maybe automatically, okay, she's a woman, memory problem, Alzheimer's disease, whereas in contrast for men, they may be underdiagnosed. And so that's what worries me in terms of some of these findings and misdiagnoses. We've also looked at differences in terms of cerebrovascular disease. And in the Mayo Clinic study of aging for individuals age 70 and older, we do see some sex differences. So the probability of white matter hyperintensities we are finding is higher in women, whereas the probability of cortical infarcts is higher in men. We don't see any difference between subcortical infarcts. And we had a discussion earlier with the HABS project because you haven't been seeing that sex differences in HABS. One of the differences, though, is that we enroll everybody from the community regardless of their cardiovascular disease. And so maybe HABS would see that if they had people, more people with vascular risk factor or vascular pathology too, but something to further explore as well. So in summary, hopefully I've demonstrated that men and women clearly have differential development in brain structure, function, and potential for pathology. And to date, it's really unclear how this affects the diagnosis down the road, how it might affect the frequency and incidence, and general development and progression of pathology. We also haven't thought about how these differences are effective throughout the entire lifespan. So, one of the problems with Alzheimer's disease in general is that it's a disease of aging. And so there are very few studies right now that have looked at, changes over puberty, changes in early development, and how that can set up risk of Alzheimer's disease and other neurodegenerative diseases as well. So next I want to, or lastly, talk about some of the sex and gender differences and risk factors for Alzheimer's disease. And I want to think about this a little bit differently. So a lot of times when we're thinking about risk factors or we're thinking about incidence, it's either men have more or women have more, or men are at greater risk or women are at greater risk. But I think we're at a stage in the field that we really need to move beyond that and start to think more of the effects of what some of these sex differences are. So for example, there are a lot of different ways that there can be sex differences in a variety of risk factors. So one is that you have the same frequency among men and women, but they have different effect. And in the past, studies have suggested that women who are E4 carriers are at greater risk of developing Alzheimer's disease compared to men. But the frequency of the E4 allele is the same in men versus women. Another way a risk factor can be different is that they have the same effect, but a different frequency. So if you think about low education, and even with smoking or with exercise, there may be some things with exercise, so let's stick with education and smoking. Generally, it's been found to have the same effect on the brain for both men and women. Typically don't see different effects there. But historically, women have had lower education, and so when you think of the total attributable risk, there are more women with lower education compared to men. Third, there are risk factors that have both different frequency and different effects. And I think a good example is with head trauma. So in general, men have a higher frequency or prevalence of TBIs, but there are some recent studies coming out, especially looking at athletes, high school level athletes, and even some college athletes, particularly with soccer players, where they're finding that the effect of heading in soccer is more detrimental for women compared to men. And so that's a case where TBI may be more frequent in men, but it may have more detrimental effects in women. And then lastly, of course, there are risk factors that are restricted to one sex. So for women, oophorectomy, removal of ovaries, pregnancies. For men, prostate cancer is an example. So before I get into some specific or variables that meet some of the other criteria in terms of differential risk for men and women, typically when you hear about sex differences and you hear about risk factors, the first thing that comes to play is always, well, if there's a sex difference, it's got to be hormonal. And it's absolutely true that hormones play a huge role in a lot of these disease processes, but there's also more than that. So clearly, as I'm sure everybody knows here, with menarche and with puberty, there's increases in both testosterone and estradiol in women. But of course, in women around menopause, you have the dramatic decrease in estradiol, whereas men, there's typically a slower decrease with testosterone. Similarly, both men and women have, women have testosterone, and that seems to be relatively stable, although decreases towards later ages. And men have estradiol, and that's also relatively stable over the age group. So how can some of these hormonal changes possibly affect risk of neurodegenerative diseases, including Alzheimer's? Well, estrogen is a critical signaling molecule in the brain, and the estrogen receptors are widely distributed in the brain for both men and women. Interestingly, the estrogen receptor ratios change with age in the hippocampus, and this may differ by sex as well. There has been a lot of really good research by Roberta Diaz-Britton, who has looked at the effects of estrogen and estrogen decrease over menopause on risk of Alzheimer's disease. And her theory, and I'll get into hormone replacement in the next slide, is that there have been some studies that have suggested that hormone replacement therapy given several years after menopause is detrimental, whereas if you give it around the time of menopause, it may be beneficial or at least not detrimental. And so her hypothesis of this is that if you give estrogen to neurons that are healthy, it can be beneficial and actually prevent amyloid pathology. But if the pathology is already starting to develop and then you give estrogen, it can enhance and be detrimental down the road. So in relation to menopause, fits in with the idea that if you give estrogen soon after menopause, then that may be beneficial or at least not detrimental. And so building on this is just a summary of studies that have looked at hormone replacement therapy, again, suggesting that premenopausal or early menopausal may be protective, whereas late menopausal may be detrimental. Now, there's still a lot of questions in terms of hormone replacement therapy, and I would never suggest starting hormone replacement therapy to prevent Alzheimer's disease or other neurodegenerative diseases. The most recent study that came out with hormone replacement therapy was the KEEP study, the Cronus Early Estrogen Prevention Study. And that study randomized women to either conjugated equine estrogens, 17-beta estradiol, or placebo, and all women had to be between 6 months and 3 years of menopause. And there was a 4-year treatment phase, and they followed them up for another 4 years now, so 8 years. And to date, they're not seeing any differences in cognition between any of the groups. So it's not detrimental to cognition, it's not necessarily beneficial. Most of these women are still young, in the 60s, and so we don't know what the long-term effects are. But I think the huge benefit of that study was that they did show, of course, that hormone replacement therapy significantly improved mood, it improved sleeping, and it decreased hot flashes. So I think the main point, one of the main messages that came out of that paper was that, okay, may not be seeing beneficial effects on cognition, but there are a whole lot of other factors that affect quality of life that women are receiving benefits on. The other thing that has started to come out of that study that at Mayo we've been looking at a lot more is that the type of estrogen that's delivered may matter. And so in that particular study, we did find differences between 17-beta estradiol and conjugated equine estrogens, specifically when you start to look at changes in the brain. So Kajal Kantarsi had imaged the women at the Mayo site, which was just over 100, at baseline, the 4-year point, and then 8 years after randomization. And interestingly, she saw that there were subtle changes, actually slightly detrimental, although not significant, for those women that were on both estrogen therapies. But as soon as it stopped after 4 years and they followed them up, those changes went away. But the worst changes were with conjugated equine estrogens. 17-beta estradiol seemed to be a little bit more protective. In addition, she also imaged all the women at the Mayo site at 8 years post-follow-up. And although we only had 30 placebo, 30 conjugated equine estrogens, and 30 17-beta estradiol, she did see that those on 17-beta estradiol had lower amyloid levels compared to the other two groups. And it was even more striking because the 17-beta estradiol group happened to have a much higher frequency of ApoE, so you would expect them to have higher levels of amyloid. And in fact, the conjugated equine estrogen group had higher levels of amyloid rather than lower levels. So this suggests to us that the type of estrogen matters and may be important going forward. And so she was funded recently to look among the entire KEEPS cohort to see if the study can be replicated. So with work that we're doing going forward, we're trying to focus a little bit more on 17-beta estradiol, but really to also compare these different estrogen concentrations. In relation to estrogen as well, some of the work we're doing is looking at the effects of bilateral oophorectomy prior to natural menopause. So women that have an abrupt endocrine dysfunction and rapidly reduced estrogen prior to their natural menopause, particularly at earlier ages, are at increased risk of developing both cognitive impairment and dementia. So particularly the age of 45, if you go up to 48, if you go up to less than the age of 45, the risk is even higher. And Walter Roca has done some work looking at comparing those that were on estrogen replacement therapy. And so if you have a oophorectomy at 45 and they're on estrogen replacement therapy up to the time of natural menopause, their risk is reduced slightly, but it's still significantly higher than a woman that went through natural menopause. Another question that comes up with sex-specific risk factors, and particularly focusing on women, is the effect of pregnancy. And there's been a lot of interest of late and a lot of discussion at the last Alzheimer's International Conference this past summer. Most of the studies have suggested that the number of pregnancies, so a higher number of pregnancies, are associated with a reduced risk of Alzheimer's disease and dementia, although there was recently a study out in neurology that said the opposite. There's always opposites, but... and so, the question came up, okay, what is the mechanism? So women that have more pregnancies are at reduced risk of developing Alzheimer's disease. And the first thing that came up, as I mentioned, tends to be hormones. So when women are pregnant, they have much higher levels of estrogen. And so maybe these high levels of estrogen are protective to their brain or in terms of long term. But I think that can be one mechanism, and I'm sure it plays a role, but I think there's also other mechanisms that we need to start thinking about. I've worked with a nephrologist looking at preeclampsia and cognition, and what I've really learned from her is that pregnancy is a stress test. And so if you get through pregnancy, you don't develop gestational diabetes, you don't develop hypertension, you don't have other complications, you're going to end up being more healthy than somebody that does. And maybe prior to pregnancy, you have two people, neither of them are showing any risk factors, but pregnancy can unmask some of these underlying symptoms. And so, another reason that fewer kids may be associated with risk is that maybe they had a difficult pregnancy to start with. Maybe they had preeclampsia in their first pregnancy or first and second pregnancy, and they're less likely to go on to have other children. So again, I think hormones should be in the discussion, but I think we also need to talk about a lot of other factors in terms of the relationship between pregnancy and dementia, changes with inflammation and vascular. So we've started to look a lot more at the role of hypertensive pregnancy and brain changes long term. In the past, most of the research has been done within about 5 years after preeclampsia, where they look at white matter changes. And so we've started to look at more of the long-term effects. And in the Family Blood Pressure Project, we did find that hypertensive pregnancy disorders were associated with worse cognition and lower brain volume a mean of 60 years. And with the Family Blood Pressure Project, that included both Caucasians and African Americans, and we did see the same association among both races as well. So one of the, I've talked a lot here about sex-specific risk factors and have talked a lot about women. There's not as much in terms of developing sex-specific risk factors for men. Or even looking at sex differences and finding risk factors for men. In the Mayo Clinic study of aging, we have started to look more closely at some of these risk factors for sex differences in some of these risk factors. And this was an initial paper a couple years ago led by Shane Pankratz from our group, who looked among individuals 70 and older, could we find different risk factors for men versus women? And again, what you can see up here, both education, memory concerns, stroke, and AFib were both significant risk factors for men and women. Women had more midlife cardiovascular risk factors. And for men, the only two factors that came up was BMI greater than 30 and never married or widowed. So I think we need to do more work down the road trying to identify risk factors for men as well. The never married, widowed is consistent among other studies. And one of the reasons for this, and this is very probably sexist to say, but I'll throw this out there. One of the hypotheses for this is that in the past, historically, it's the women that have generally taken care of the families, that have generally made sure husbands have gone to the physicians, children have gone to the physicians, in general. And so the thought right now is that men that are elderly, 80, 90 plus, if their wives have died, for example, then there's less likely to have somebody that tells them to go to the doctor or get regular checkups or whatnot. And so, it's possible that single men then are at greater risk. Again, in terms of looking at and trying to identify risk factors for men, we did find a difference in the Look Ahead study. And the Look Ahead study was a clinical trial of overweight or obese diabetics aged 45 to 76 that were randomized to a 10-year multidimensional behavioral intervention. And cognitive impairment, either MCI or dementia, was adjudicated about 10 to 13 years after enrollment. A large sample size, 3,800 individuals, about 60% of women, but a pretty good difference. Interestingly, there were no differences in terms, sex differences in terms of treatment response. But what they found was that cognitive impairment across the age range was much greater for men as compared to women in this study. So this is a little bit different than what we were, actually, when we found this study, I worked with Mark Espeland on this. We were kind of taken aback because we figured that diabetes would be a greater risk factor for women compared to men, given the changes with metabolic syndrome and past research suggesting it may be more of a risk factor for women. So we did a whole bunch of studies in the paper, a lot of sensitivity analyses trying to get rid of this finding, and it stuck. So this has led to some additional research that we're currently looking at more in terms of frank diabetes and risk of dementia, as well as looking at potential sex differences in terms of diabetic medications and how that can differentially affect cognition as well. So lastly, I just want to touch on, I've talked more broadly, but some of the work that we're doing at the Mayo Clinic to start look at sex differences, both sex-specific as well as, again, differences in terms of risk factor. So one of the grants, RF1 funded, we're just about a year and a half in. We have about 3,000 women that have been enrolled in the Mayo Clinic study of aging. And I mentioned earlier that we have the advantage of something called the Rochester Epidemiology Project, where all the medical records within Olmsted County healthcare providers have been linked since 1966. So the goal of this study is to look back through all these records and abstract information on pregnancies, including whether they were hypertensive, glucose dysregulations, and other factors, gynecological surgeries, all birth control, hormonal therapies, as well as things like tamoxifen and other breast cancer-related therapies. And then subsequently look at the effects of risk of Alzheimer's disease, so both Alzheimer's pathology, and we can also look at cerebrovascular disease because we really don't know yet whether there's more of an effect of hormones on vascular disease or Alzheimer's disease, as well as cognition. We've also just started incorporating hypertensive pregnancy questionnaires and menopausal symptoms into this. So it's been extremely eye-opening if you've ever looked at written medical records and actually going back through all these records and trying to read it and everybody disagreeing on what the medication is actually saying. But I think this will give us a great opportunity to really look at the lifespan and hormonal effects over the lifespan for these women and what the long-term effects are. It's also interesting to look in this cohort because we do have some women that were on Enovid when it first came out, which is the first birth control pill, which had very high levels of estrogen and progestin. And those levels, of course, have varied over time. So this will also give us an opportunity to look at high levels versus lower levels of hormone therapies. In addition, we are looking among men at the effects of prostate cancer and subsequent cognitive decline, particularly whether androgen deprivation therapy is associated with cognitive decline. So in our initial analyses, we do see an association between androgen deprivation therapy and worse cognition. However, once you correct for age, it's no longer statistically significant. We also have, although orchiectomies are not common anymore, going through the Rochester Epidemiology Project, there have been past investigators that have established orchiectomy cohorts. And so it'd be really interesting to look among those cohorts and see what the subsequent risk of dementia, stroke, a variety of neurodegenerative diseases are as well. And then lastly, we were recently refunded for our Specialized Center of Research Excellence on Sex Differences. And our focus here is to look at abrupt endocrine dysfunction, aging, and Alzheimer's disease. We are initially focusing on the effects of premenopausal bilateral oophorectomy, which contributes to abrupt endocrine dysfunction, and the various aging processes that may contribute to cognitive and functional aging. Now, I haven't given you a lot of background slides in terms of the work done at Mayo, particularly by Walter Roca with bilateral oophorectomy. But there have been some recent studies that have suggested that women who have premenopausal bilateral oophorectomy, especially before the age of 45, may have accelerated aging in terms of epigenetic changes and multimorbidity. And so we're trying to look at that a little more closely, and our plan is to recruit 500 women, 250 with and 250 age matched without a history of premenopausal bilateral oophorectomy. We'll bring them in for about a 6-hour day of testing, including cognitive tests, functional tests, body composition test, a variety of aging-related markers that we're working on with our COGOD Aging Center, and look to, as well as with neuroimaging, both MRI and amyloid PET, to see if there are differences in those women that did have premenopausal bilateral oophorectomy and those that didn't. Interestingly, for some of our preliminary data for this, it turned out that there were about 80 women that are enrolled in the Mayo Clinic study of aging now that were also initially enrolled in this oophorectomy cohort. And even among these 80 women, we are seeing, who have about a mean age of 60, I believe, we are seeing differences in frailty. So the women that had premenopausal bilateral oophorectomy have a greater number of comorbidities and more markers of frailty than women that did not. So we'll follow them up and do a lot more in-depth testing. And then we also have an animal project trying to understand the effects of estrogen after orchiectomy, the timing of the estrogen on cognitive and functional decline. And our goal down the road is to eventually try and understand these mechanisms, but not only focus on bilateral oophorectomy, focus on a lot of other endocrine disruptors that are present in both men and women, determine how it affects the aging processes, including Alzheimer's disease and dementia, and try and identify means for interventions. So lastly, just in summary, going back to the first part of the talk, I think it's pretty clear that, when we talk about burden, more women than men have Alzheimer's disease. There are more women affected at a given time. There are more women that are more women at older ages. But the prevalence and incidence are not so clear and more likely equivocal. So, again, as a field, and I mentioned earlier, it's kind of my bugaboo where I think we really need to be careful about how we present this, both for the lay audience in order to cause excess worry, but also in terms of presenting to primary care practitioners, so less likely to have misdiagnoses. There are clear evidence in terms of sex differences in brain structure. We're still unclear exactly how this relates to the clinical phenotype, but I think this opens a wide avenue, and I know Prashanti Vermuri and a lot of others are looking at differences in white matter tracts and between men and women and how this might affect susceptibility or the showing of clinical symptoms for the same degree of pathology. There are some risk factors that are sex-specific, but many risk factors are different in men and women. And in the past, the majority of research that has been done today typically controls for sex, because it's a lot easier to adjust for sex than to try and deal with all these differences. And we had a talk earlier, and once you start thinking about sex differences, there's a whole bunch of other things to think about. But hopefully I've at least started to convince you that it is important to look at these sex differences. Not only in humans, but in also many of the animal models that you're working on. So, right now, a lot of times we hear about individualized medicine, or precision medicine, but sex and gender aren't often incorporated in that. And so, really, in this area that we're moving forward, that's one of the first things that I think people should consider. Sex and gender, not only for Alzheimer's disease, but for a variety of other diseases, again, in terms of risk factors, clinical presentation, and treatments. So I've had a great opportunity to work with several, both at the Mayo Clinic, as well as in Chicago and Sweden. And so I'd like to thank you for listening to me for a while, and I'm happy to answer questions.
So we'd be happy to take questions. And Ray, you're here with the right here. Excellent. And any questions for Michelle?
Yeah, hi. Very interesting talk. I'm a lay person. I'm trying to find out if somebody has a diagnosis of MCI, what's the difference if the doctor is telling you that you don't have memory issues but executive function? What's the difference in the two?
So, great question. I'm not a clinician, so if there's a clinician, please feel free to stand up and correct me. There are different types of cognitive changes and different types of domain. So cognition can be language, it can be memory, it can be kind of planning different things out. And so with different neurodegenerative diseases, different parts of those cognitive domains are affected. So something called primary aphasia, your language is going to be affected, but your memory will be the same. Does that help? Oh, Risa, you can answer that better than I can.
So really great. I'm so sorry I missed the first part, but hopefully people will fill me in. I had a question about, I haven't read that Mark Espelin study that you worked on, and in the diabetics, were the men and women equal in the terms of the amount of vascular pathology they had for their given diabetes profile? Because as we were talking about earlier, the question about whether,
Yep.
the vascular component is different in men and women, but in that study, are they equated for that?
They were the same. Also, same frequencies of severity of diabetics. We didn't see a sex difference in terms of type of diabetic medications. Again, what ended up coming out is that, okay, well, maybe women were more likely to be taking hormone replacement therapy, and so that protects them. It's kind of the last straw. But no matter what we did with that study, we couldn't get rid of that effect.
Thank you, great talk. So my question would be about the white matter that you are referring that is more in men than in women. I know there's only some small regions in adult brain of, showing plasticity, but there are, so have you, or do you know whether someone had looked into white matter with possibly overreactomies or such things? Do you know something?
So, well, actually, women had more white matter hyperintensities than men did in our study. And so, and the effect of overreactomies, premenopausal bilateral oophorectomies, that hasn't been looked at yet. And so that's what we're hoping to look at in some of our studies going forward. There's a lot of interesting research looking at sex differences in terms of diffusion tensor imaging and white matter infrastructure. And it does seem like there are sex differences there as well. I'm not going to go into specifics, that's Prashanti Murray's work, but some interesting findings there too.
Thank you for a wonderful talk. And my question is also about white matter. You said that men had more white matter than women. And a very old, dim memory of mine is from one of the early gender studies on neuroanatomy, which was that the corpus callosum was thicker in women, was bigger in women. And I don't know if that has been largely fallen into disfavor, but that sort of is more compatible with your model of men having greater laterality of language and women having a better bilateral distribution. And another quick question is whether you could comment on the gender difference in Parkinson's disease, which tilts in the male direction.
Okay. So with your first question with the corpus callosum, I can't really comment on that. I'll have to look back in the literature and take a look at that. In relation to Parkinson's disease, yes, so men have 2-3-fold greater risk of developing Parkinson's disease than men. Or than women. And with Silke and earlier we were talking about some of these differences, and I don't think we don't fully understand that. One of the hypotheses has been that men are more exposed to pesticides, working in the fields, those type of occupations, but clearly that's not everything. And I think Silke has a really nice mouse model and is looking at differential effects of estrogen, and that could be important down the road as well.
Thank you. I think you said that women have a high percentage of typical presentations than men, and that had implications in terms of misdiagnosis, or at least the atypical ones. But I don't have a sense, and perhaps you could help us, what's the magnitude of the difference between men and women in terms of typicality or atypical presentations? Is it small or is it substantial?
That's a good question. So this was really the first study that, to my knowledge, had looked at that in terms of autopsy samples and actual pathology. I haven't really seen other studies. Again, sorry, Risa, I'm referring to you, neurologists, if you have a general idea of what that might be.
I think there's a sex difference. There are some studies with higher progressive aphasia are asking about sex differences in PPA, but I haven't seen it in like the PCA, the visual,
Right.
Whether men have more visual and women have more verbal, since we tend to be more verbal. I don't know.
I mean, based on that, the neuropath finding, it would be interesting to go back in some of these studies, even with HABs and with MCSA, where we have amyloid PET, tau PET, and start, now that we have some evidence of the pathology, start taking a look at this more and which cognitive symptoms end up coming first for men and women.
I have two questions. One is about obesity in men, and have you looked at testosterone levels and whether they're lowered in obese men, and could that be a risk factor? And the second question is around the bioavailability of in the menopausal HRT studies and MRI, and is there a differential bioavailability of the 17-beta estradiol versus the equine?
For your first question, no, we haven't looked at testosterone levels and the effects on obesity, but that's a great idea. I think it would be really interesting to run a whole panel of steroids, look at androgens, testosterone, even estrogens in men and women that are obese. Secondly, in terms of bioavailability, I don't know what the answer to that is. The 17-beta estradiol is typically in a patch. And so I don't know if that necessarily affects the bioavailability differently or not. Talking with our endocrinologists in the Women's Health Center who work primarily with menopausal women and determine the right hormone therapies, one of their big concerns is that, typically or clinically right now, the hormone replacement therapies that's suggested is typically, it's elevated compared to having no estrogen levels, but it's not typically, they don't know what the correct physiological dose is. And so they're doing a lot of research right now thinking that, well, in a lot of these women, maybe it should be higher than, what it currently is. Whereas in other women, it might be lower because depending on your certain SNPs that you have, you metabolize estrogen differently. So I'm kind of adding more to the confusion than answering it. But I think that's a big question and something that needs to be looked at a lot more. Again, just because they don't know what the best physiological dose or the way to administer it is.
That was a wonderful talk. Thank you so much. I have two questions also, and one was, and you may have said this, but is there any evidence for sex differences in very early stage pathology? I mean, has it looked at for HAB or for the Diane studies, that sort of thing? That's my first question. And then the second question is, do you have any ideas or thoughts about mechanisms for the sex difference in ApoE4 carriers?
Okay, so the first one in terms of early stage pathology, several of the studies, probably with HABs and what we've seen with the Mayo Clinic study of aging too, it has been in cognitively unimpaired individuals. So we are at the earlier stages now. What the earliest stage is, I think will be a little bit difficult. But even among cognitively unimpaired individuals who are amyloid positive, we're not necessarily seeing those differences. Now, we did have a talk about the Columbia Kindred, where is she? Yes. And so in their, you can explain this better, so I'm sorry if I butcher this, but they are seeing sex differences in their early onset AD. So, again, not sure exactly what's going on there, and that's the first I've heard. I don't even know with Diane, with their early onset AD, whether they're seeing sex differences. So, but in general, in the general population, no, we're not.
So when you say sex differences for Diane or the Colombian kindred, are you talking about amyloid imaging?
Yes.
In particular?
Yes. Yeah, but whether we look at amyloid imaging or CSF, we're really not seeing a whole lot.
Okay, great. And then the second question was just about ApoE4.
Yeah, I don't know. Actually, I think, and we were talking about this earlier too, a lot of studies that are coming out now aren't seeing as big of an effect of the sex difference for women. So women with ApoE4, it may not be as big as what the first studies, FAR study in the late 90s had shown. I've seen, we had a special issue on sex and gender for Alzheimer's and dementia, and I guest edited that, and the first 6 papers were on ApoE, sex, and a variety of outcomes, whether it was cognition, imaging, some were longitudinal, some were cross-sectional, There were 3 that included ADNI data, which was interesting to compare. And really, the data was all over the place. There are some times where it looks like ApoE was a greater risk factor for women, sometimes for men. I think overall, there probably is a slightly greater risk for women, but it may not be as big as what we thought. So then in terms of the mechanism, at first we got to figure out whether there is a difference or not and what the difference might be.
How about if we just take 2 more questions if there are 2?
Thank you, that was a great talk. Excuse me. Could you comment more on the modifiable lifestyle risk factors for Alzheimer's, including low educational attainment, lack of cognitive stimulation, which is thought to be globally the largest modifiable risk factor for dementia, or for Alzheimer's, and exercise, which is the lack of physical activity, is thought to be the #1 risk factor in the U.S. And it really seems like women, both globally and in the U.S., have lower levels of educational attainment, less cognitively stimulating jobs, and traditionally did not exercise as much as men.
Right.
So I would be interested in your comments on that.
No, I think that's certainly I agree with exactly what you were saying, and that may be it'll be interesting to look down the road 20, 30 years as women do have more education compared to beforehand. Women still aren't exercising as much as men, at least based on the U.S. surveys. But to look at these cultural changes over time, I think is going to be really interesting. Now, whether it actually affects resilience or it affects the pathology is questionable, but we'll have to see whether it does have an effect or not. One thing that's where a little bit I sometimes think that Europe is ahead of us. They're doing a lot more of the behavioral intervention trials, the finger study, and now we are doing that in the U.S. But they do get good effect sizes, and so I think it is important and something that we need to push.
Is there one more question? No, I think we're all set. So Michelle, thank you very much.